being that myofibrillar myopathy is a disease caused by an error in the genetic code. The only real treatment or cure can only come from some form of gene editing or gene therapy, which isn't currently available. But for now, we focus on therapies that can slow progression and preserve muscle. Autophagy. It is the body's cellular recycling system. The word comes from the Greek words auto, meaning self, phagein, meaning to eat. Literally, it translates to self-eating. While that sounds destructive, it is actually a vital survival mechanism. It is the process by which cells clean house by breaking down old, damaged, or abnormal components and reusing the leftover parts for energy or to build new, healthy structures. So what does this have to do with myofibrillar myopathy? In MFM-specific proteins like desmin, alpha-biocrystalline, filament C, or BAG3 mutate and misfold. These misfolded proteins clump together into aggregates that physically clog the muscle fibers, acting like sand in the gears. Some rare genetic diseases like MFM result from a total lack of ability to clear a specific protein. While autophagy can't fix the gene, upregulating it can help manage the symptoms by clearing the backlog of trash. Increasing autophagy is currently considered one of the most promising therapeutic strategies for myofibrillar myopathies, MFM, and other protein aggregate myopathies. In diseases like MFM, the problem is not just that proteins are clumping together. It is that the cellular trash can, the ubiquitin proteasome system, is overflowing. When that system fails, autophagy is the only backup mechanism capable of swallowing and digesting these large, sticky protein clumps. Autophagy is the cleaning crew. The cell identifies junk such as damaged mitochondria and misfolded toxic proteins that clump together, forming aggregates. The cell forms a membrane around this junk. This bag of trash is called an autophagosome. The autophagosome fuses with a part of the cell called a lysosome. The lysosome contains enzymes that act like a trash compactor and acid bath. The junk is broken down into basic raw materials, amino acids, fatty acids, sugars, and released back into the cell to be used as fuel or to build new healthy cell parts. Research has been highly successful in animal models, mice, and zebrafish for several types of MFM. Desminopathy. In mouse cells expressing mutant desmin, stimulating autophagy, cleared the aggregates, and reduced cell death. CRYAB. Alpha-biocrystalline mutations in mouse hearts with this mutation. Increasing autophagy cleared the toxic protein clumps and slowed heart failure. Conversely, blocking autophagy made the disease significantly worse. BAG3 myopathy. In zebrafish models of BAG3 myopathy, researchers found that the protein recycling system was stalled. Reactivating it helped clear the damage. Blocked pathways. In some specific genetic forms of MFM, like certain filament C mutations, the machinery required to perform autophagy is itself broken. In these cases, trying to step on the gas might not work because the engine is missing parts. Autophagy is always happening at a low level, but the body cranks up the volume when it is under stress. The most effective ways to induce autophagy include fasting. This is the most potent trigger. When you stop eating for an extended period, usually 16 hours in humans, insulin levels drop and the body perceives a nutrient shortage. To survive, it begins eating its own damaged parts for energy. Caloric restriction. Consistently eating fewer calories than required without malnutrition keeps autophagy active. Ketogenic diet, high-fat, low-carb diets mimic the metabolic state of fasting ketosis, which can trigger similar autophagic pathways. There are no pharmaceutical drugs FDA approved specifically for the purpose of increasing autophagy. Researchers have identified several existing FDA-approved drugs that induce autophagy as a side effect or secondary mechanism. 2 existing drugs have shown specific promise in translating this to human MFM treatment. Metformin, the diabetes drug. Surprisingly, this common diabetes drug is currently a leading candidate for treating BAG3-related myofibrillar myopathy. Metformin activates an enzyme called AMPK, the cell's fuel gauge. When AMPK is activated, it signals to the cell that energy is low, which suppresses mTOR and turns on autophagy to generate fuel. A major clinical trial called the TAME Targeting Aging with Metformin trial is being designed to see if metformin can delay age-related chronic diseases in humans. Using the tiny zebrafish, Associate Professor Robert Bryson Richardson from the School of Biological Sciences and his team of researchers were able to show that metformin removed the accumulating damaged protein in the fish, prevented muscle disintegration, and restored their swimming ability. Real-world example. There are reports of compassionate use of metformin in children with BAG3 myopathy. It is showing positive clinical stabilization. The research conducted by Robert Bryson Richardson and Avnika Ruparelia has given hope to boys with BAG3. Compared to previous case studies, the progression of the disease has been slower, likely due to metformin. Given that metformin is taken by millions of people for diabetes and known to be very safe, this makes clinical translation highly feasible. And in fact, many patients are now taking it. Rapamycin, sirolimus. This is the most potent and well-known autophagy inducer. Its original use is an immunosuppressant used to prevent organ rejection after kidney transplants. It is considered the gold standard for longevity research. It has successfully extended the lifespan of mice and other animals in lab trials. It works by inhibiting a protein called mTOR, mechanistic target of rapamycin. When mTOR is active, usually when you eat, it shuts autophagy down. By blocking mTOR, rapamycin tricks the cell into thinking it is starving, which triggers the autophagy recycling process. While direct large-scale trials for MFM are rare, rapamycin has been tested in a phase 2 trial for inclusion body myositis IBM. IBM is very similar to MFM in that it involves protein aggregates in muscle. The trial showed that rapamycin could stabilize the disease and prevent the loss of walking distance compared to a placebo. This suggests the mechanism is valid for protein aggregate muscle diseases. The double-edged sword warning. While increasing autophagy is beneficial, it must be balanced. Excessive autophagy is actually a cause of muscle atrophy, wasting away. If you turn it up too high, the muscle eats its own healthy tissue. Summary: the consensus in the scientific community is yes. Therapies that safely and controllably increase autophagy, like metformin or rapamycin, are currently the best hope for treating the underlying cause of myofibrillar myopathies, rather than just managing the symptoms. Other research unrelated to autophagy. CDD-2107 is an investigational botanical drug developed by the Center for Chinese Herbal Medicine Drug Development at Hong Kong Baptist University to treat myofibrillar myopathy. It is derived from Chanamelis fructus, a traditional Chinese medicine commonly used for joint and muscle disorders, refined using modern extraction techniques, specifically aimed at treating BAG3-opathy, a form of myofibrillar myopathy, designed to improve muscle strength, enhance mobility, and increase independence for patients. The drug has obtained the Orphan Drug designation from the U.S. Food and Drug Administration (FDA). It is the first botanical drug in Hong Kong to receive this qualification. As of late 2025, it is in the pipeline with plans to submit an investigational new drug (END) application to the FDA by early 2026 to begin clinical trials. Thanks for watching.